So thanks so much for that introduction, and thanks to both Dayu and Tiago for inviting me. This meeting has been really great. I also really want to thank Kristen. That was a spectacular introduction to machine learning and its application to behavior, and it frees me to not have to explain a lot about supervised or unsupervised learning. For me, this meeting has been really, really useful in terms of helping to continue to clarify my own thinking about what characterizes a reasonable behavioral representation. I'll just mention that Kristen and I, along with some others, are in the midst of writing a review, and I mentioned this publicly because it means that we'll have to finish this review at some point, right? We're like, now we're on the hook, basically. But these kinds of conversations have been incredibly useful for us because I think we collectively are trying to advocate for some positions about how we should be thinking about, or at least suggest some ideas about how we should be thinking about what makes for a good behavioral representation, and really talking to groups of people like you is really useful in terms of guiding our thinking. I speak very quickly. I have 45 slides. Left to my own devices, I can get through this talk in 25 minutes. But I've seen what this audience is like, and I know that I'll be interrupted constantly. I'll just mention that I have 2 slides of data that are at the very end that I really want to get to. So if I can try to get to the end, I would if you let me get to the end, I'd appreciate it. But I don't have very many slides, and I can go very quickly. All right, so let's just get started. All right, so this guy is a deer mouse foraging for food in the wild. And this is a task that depends crucially on this animal's olfactory system. So if you do the kind of experiment that my lab likes to do, like Tiago mentioned, which is to make recordings of this animal's piriform cortex, what you'll find are robust representations of whatever the animal is smelling at any given moment in time. Now, the piriform is part of a highly recurrent circuit, and so it's easy to see information that kind of echoes smells this animal has encountered in the recent past. And there's definitely, just because of the circuit configuration, some form of predictive coding going on here. And so with the right lens, you can discern something about what the animal believes it's going to smell in the near future. Now, somehow, this animal's brain has to sort through all of this temporally admixed information, not just about smell, but from all of its senses, and alight upon just the right behavior to express at any moment in time to let the animal reach its ethological goal, which in this case is to find food and ultimately to survive. And things out here in the real world are tough. They're not like they are in the lab. So it's not enough for this animal to kind of do one of these cycles of sensory motor coupling in order to reach its goal. Out in the real world, animals have to kind of traverse these cycles tens, hundreds, or thousands of times, often in prescribed sequences, in order to solve their kind of ethological existential problems, to find food, to avoid predators, to obtain shelter and mates, and so on. And so this is really the core question that my lab works on. We want to understand how sensory cues that are often in the world organized on timescales of single to tens of milliseconds inform the brain and allow the brain to compose complex patterns of behavior that themselves are, as I'll show you, often organized on timescales of hundreds of milliseconds to allow animals to reach ethological goals. We're also, of course, interested in how these actions then change the way that animals interact with the sensory world. And we've become recently very interested, although I'm not going to talk about this at all, in how the brain might jointly represent sensation and action to facilitate predictions. So this problem, of course, is a large problem. It spans many areas of neurobiology, and I think it arguably covers most of what the brain does. And so we've made, as Tiago mentioned, a kind of simplifying assumption, which is that we focus on what is arguably ethologically the most relevant sense for most animals, and that is, of course, the sense of smell, as Lisa mentioned earlier in this meeting. And so we're interested in how ethologically relevant odor cues drive actions and how those actions inform the ability of the animal to interact with the olfactory world. So when I started my lab, we had some pretty naive ideas, right? I think the ideas were like, we'll take some mice, we'll get some ethologically relevant odorants, we'll throw the mouse in some sort of arena, we'll expose the animal to the odorant, amazing innate behaviors will emerge, and then we'll use the sorts of mechanistic tools that Susanna and others have developed to take apart circuits and discover how the brain works. And what we quickly learned was even the presumption at the very beginning that we could understand how odors influence behaviors was wrong. When we kind of surveyed the available set of tools for tracking behavior and for understanding behavior, and we compared it to the richness of the behaviors we were observing by eye, there was this huge mismatch. We didn't really, at the time the work that I'm about to describe to you began, we didn't really have any quantitative or comprehensive access to the underlying structure of behavior, particularly in untethered and untrained rodents, which is the thing that we cared about. We wanted to understand the kind of baseline pattern of action that mice in the lab exhibit, and we wanted to understand how smells change that pattern. And we didn't even have access to the kind of the first part of this. And so just to give you a sense that this kind of claim that the tools don't quite match the problem, I'm going to give you a very, very simple example. And this is the simplest possible example in behavioral neuroscience. What you're looking at here is, of course, a mouse in an arena called a circular open field, which obviously is just a bucket. And we're going to look at this mouse with a $10 webcam that I've placed above the animal. And this is actually a very typical configuration for a behavioral neuroscience experiment. So if you play the movie of this mouse kind of wandering around, even those of you who have never, ever seen a mouse, I think will quickly appreciate that this animal's behavior is actually highly organized and structured. It's not behaving randomly, but rather its behavior is evolving in a kind of structured and arguably predictable way that allows this animal to reach an ethological goal, which in this case is to gain information about this new little universe, this blank universe, but a universe that's new to it all the same, and to understand what's in its environment. But the tools that were available to us when we began this work were essentially video-based point tracking software, right? So we can measure, at the time the work began, those tools were fairly crude, sort of the sorts of tools you could buy from Noldus. And now, of course, those tools are much more sophisticated through the work of Kristen and the development of deep learning-based approaches. We can now track many identified points fairly trivially on this animal. So we can identify sort of where the mouse is in the bucket. We can use that information to understand its velocity, its position with respect to the wall or the center. We can look at the angle of its head, the position of its tail, and so on and so forth. And if you have particularly ambitious graduate students, as I do, they can make these very long lists of parameters, right? Hundreds or thousands of these kinds of parameters that you can extract from sort of point tracking software. And that's basically what was typically done. And the challenge here, right, is that each one of those parameters is a kind of shadow of the thing we care about, which is the underlying time-varying structure of the animal's behavior. And we actually talked about that a little bit in part of the discussion in Kristen's talk. And so in order to deal with this, my laboratory has adopted an ethological hypothesis, and it's a hypothesis about behavior. And that hypothesis is that this animal's behavior, like that of all animals, is composed out of a series of behavioral modules, right? And so in this case, each behavioral module is going to be a stereotyped, repeatedly used, three-dimensional motif of action. So those are, in the case of the mouse, those are going to be things like the first half of a rear, or step to the right, or turn to the left. And that the animal's brain takes advantage of these modules and places them in sequence quite flexibly to allow the animal to compose dynamical patterns of action that let the animal explore the universe in a nearly infinite variety of ways. Okay, and so for the last several years, my lab has been seeking to develop machines that combine 3D machine vision approaches with unsupervised machine learning techniques, which you've heard about, in the hopes of being able to discover some sort of underlying modularity in mouse behavior. And the idea is that if we can identify this modularity, we will not only be able to better describe behavior, but presumably we should be able to better predict behavior. We should be able to predict an animal's future behavior based upon its present behavior. And as I'll show you in the second half of this talk, we should be able to predict what's going on in the animal's brain based upon the animal's ongoing pattern of action.
How would look data which would prove this hypothesis is false?
How would you, there's ways to prove it false. I'll show you some ways to prove it false, actually. There are ways to know whether or not, there's ways, we adopt, I will show you. There's ways to think about this. There are ways to think about it. Okay, but it is a hypothesis. Okay, so we developed a system, we call it motion sequencing, and it was first developed by a student of mine named Alex Witchko along with a postdoc named Matt Johnson. And as Tiago mentioned, this paper was published 2-3 years ago. So I'm just going to give you a high-level summary of motion sequencing and how it works conceptually. So I can spend most of the time talking about data and how we've been using motion sequencing and thinking about validating motion sequencing. So today I'm going to give you a little bit of an introduction to motion sequencing. And then I'm going to spend several slides showing a bunch of unpublished data, thinking about how we might validate a particular behavioral segmentation, falsify theories potentially about how behavior might be organized. I think there are many ways to think about that, and I'm going to advocate for one way of thinking about it. It's not the only way, but it's a way to think about it. And then I'm going to talk a little bit about predictions, and I'll end with some really exciting unpublished data that I'm looking forward to sharing with you. Okay, so motion sequencing, as I mentioned, takes as its input 3D videos about mice as they explore different arenas in the laboratory. And we obtain these videos by placing depth cameras, which see in 3D, above experimental arenas of choice. And so what you're seeing here, heat map, is actually the animal's height at any given point over the animal. So when this animal's head is turning red, it means the animal's rearing or sniffing. When the animal's back is red, it means its back is arched up towards the camera. And we choose to focus on these three-dimensional pose dynamics, this particular feature set, because if you think about it, an animal's three-dimensional pose dynamics do in a very coherent way, capture all of the actions the animal generates. So it's a kind of nice, balanced feature set that in a kind of unitary way comprehensively captures the animal's behavioral dynamics. We then submit these 3D pose dynamical data to a model that we've instantiated in the cloud, and which we fit in the cloud. And this model explicitly articulates this ethological hypothesis for behavior, which is that behavior is modular, it's built out of parts, and that those parts tumble one after the other in time in a relatively predictable way. Okay, so for those of you who are a little bit more machine learning savvy, I'll just mention the details. We model each one of these behavioral modules as a autoregressive process through high-dimensional pose space. We modeled the switching between these individual modules via a semi-Markov model, which affords us some temporal flexibility. We identify how many modules there are automatically without any human supervision or intervention through the use of Bayesian non-parametric techniques. And we fit the whole thing probabilistically using modern statistical machine learning techniques like Gibbs sampling. So the net of all of this is that based upon the structure of these video data alone, the inherent structure of these video data alone, you submit these video data to MostSeq, and MostSeq spits out at you essentially a parts list for behavior, the set of behavioral modules that best explains, and as I'll show you, best predicts a mouse's behavior. And because we think these modules are components of mouse body language, we choose to refer to them as behavioral syllables, and we choose to refer to the statistics that govern the transition between these syllables as behavioral grammar. So I'm not going to actually go through any of the math or the fitting procedure. I'll just refer you to this paper if you want to look at 80 pages of math justifying kind of some of the things that I've been saying. Although when I talk about how you would, I'll talk in a few slides about how we would validate such a model. But I won't talk about any more of the details of how MostSeq works. But I do want to show you some movies of these behavioral syllables to give you a sense of what I mean by a behavioral syllable, and to give you a sense that these behavioral syllables are transparent and understandable to human observers. So what you're looking at here on the left is a 3D movie of a mouse that's been placed in the circular open field. Again, heat mapped as height, so this is the mouse's nose because it's pointed up a little bit. And when I hit play, the animal's going to behave. And then at some point, a dot will come over the animal. And when that dot comes over the animal is going to generate a particular MostSeq identified behavioral syllable. And since I've looked at this movie already, now many times, I have given it a name, and that name is turn to the right. Okay, but I didn't look for a turn to the right syllable. MostSeq just identified this as a structural component of behavior. So let's watch this movie. There's some behavior. There's the dot, and during the dot, the mouse turns to the right and actually rears a little bit. Okay, and it's just going to loop, so you can see that a couple times. Is it reasonable to think of this as a kind of stereotyped unit of action? On the right are 30 separate MostSeq identified instances of this behavioral syllable being expressed, but I've drawn these from 8 separate experiments and actually 8 separate mice. And because these are all different examples, of course, what the mouse is doing before the execution of the syllable is going to be different in all of these cases. But watch what happens when the dots come over the animal. Right? So sort of in this kind of synchronized way, these mice generate this kind of coherent 3D motif of action, right? And again, this movie is going to loop, so you can see it. You can kind of get a sense of what this 3D motif of action is. Now, as you watch this movie, you'll notice that there's a certain amount of spatiotemporal variance associated with each implementation of the syllable, right? It's not always exactly the same. And that kind of spatiotemporal variance is explicitly accommodated by motion sequencing, just out of acknowledgement that no action is really expressed in precisely the same way by any animal 2 times in a row. So here is a subset of the behavioral syllables that we identify in a typical open field experiment. Using our current algorithms and cameras, we typically, in an open field experiment, will recover something like 40 of these behavioral syllables. And of course, we can look at them each after the fact and give them names, right? My favorite is this run, force, run syllable where they just kind of go, dart off as fast as they can. Each one of these behavioral syllables on average, lasts about 300 milliseconds or so. Although that is a mean. Each one of these behavioral syllables, as I mentioned, has a characteristic and syllable-specific duration distribution. So the means for any given syllable can vary from 100 milliseconds on the one end to more than a second on the other. So this is a kind of collection of automatically identified parts of behavior. How can we think about these parts of behavior being sequenced in order to generate actual behavior? Well, we can, of course, just build behavioral state maps. As is shown here. So here, of course, each node is a behavioral syllable. Its diameter represents how often in a given experiment that syllable is used. The yellow directional arrows indicate the observed likelihood of transition from one syllable to the next. And so this map, this behavioral state map, really lets us do something important for the first time in the mouse, which is to make predictions about a mouse's future behavior based upon its present behavioral state, because we know if the mouse is here, it's likely to be here in the next second or so. And actually, motion sequencing allows us to predict a mouse's future behavior with some reasonable above chance degree of accuracy many seconds into the future. The other thing that this, of course, behavioral state map affords us is a kind of comprehensive encapsulation of the overall pattern of behavior the animal adopts in any given experiment, right? At least from the limited perspective of this camera, this 3D camera. And that is extraordinarily powerful, right? It's this kind of, this kind of objective summary of everything the animal did, and it allows us to make experimental comparisons. So as we experimentally manipulate the animal, we can ask how did our experimental manipulation affect the overall pattern of behavior by comparing these different behavioral state maps in various ways. And so in published and unpublished data, we've done that in all sorts of different ways. We've done things like a very subtle optogenetic perturbations that are highly unreliable, but motion sequencing lets us understand on a trial-for-trial basis the consequence of tweaking activity in a particular neural circuit. We can, of course, take out genes. We have, for example, done a fairly comprehensive survey of autism spectrum disorder model strains and asked how an animal's overall pattern of behavior is changed when you manipulate genes over the course of the lifetime of an animal. We can make all sorts of acute manipulations. We can move animals across different experimental contexts, from the open field to the elevated plus maze to the three-chamber social assay, and ask what behaviors are in common or what behaviors are different and specific to any given behavioral context. And of course, as I'll show you, we can give olfactory cues and ask how odors drive behaviors.
So this is all in the bucket still, right?
This particular map is in the bucket, but it doesn't have to be in the bucket.
What you've been saying applies also to all
other. And we've done lots of non-bucket experiments. Yeah. So what? But the set of syllables you get, just to be clear about this, is absolutely context specific and strain specific. So the number 40 is if we image animals for 30 minutes in the circular open field of a particular strain. Like that's all. It's all specific. And if you make a change, like you move from a circle to a square, the set of syllables will be highly overlapping, but there'll be things that distinguish those two contexts. So for example, as Andres showed before, like animals love the corners, and you'll see corner-specific syllables that aren't present in the circle because there are no corners. If we change strains from C57 to 129, something like 70% of the syllables overlap, but 30% are actually strain-specific. The thing that I'm not going to talk about is that this technology affords us the ability potentially to look for genes for behavior. This is a project that Hopi Hoekstra and I actually have. We've been working on together for several years. It provides, because it's unsupervised and doesn't depend on a prior definition of behavior, we can apply it to all sorts of different contexts and understand the differences between those contexts.
In terms of predicting, can you give us an intuition how far in the future you can go and how well are you doing?
Not very, so I'm not going to show you any data for that.
It's seconds, right?
It's seconds. It's the order of single seconds, not tens of seconds. So I will say there's one. This model is unsupervised in that it identifies these behavioral syllables without human supervision. There are two key assumptions in the model and in MoSeq. One is the model itself, and I'm going to talk about that assumption in a second. Like, we assume that behavior is modular and has these switching dynamics. The other assumption, which I will experimentally defend later in this talk, because it's a reasonable assumption, is that behavior is organized at a roughly sub-second time scale. Right, and that is the time scale is an assumption. We all know that behavior is organized hierarchically on many time scales. We are specifically looking for a granular kind of organization to behavior. We are building out models now that are hierarchical that might allow us to look at longer time scale behaviors. But for now, because the model is so granular, the time scale at which we can make predictions is pretty limited. Okay?
So you get a probability of position over time?
Yes. Yes.
So how do you compare two syllables that the model spits out in different contexts, for example. Is that just by observation, or is there some quantitative method to...
Yeah, so again, the model identifies an autoregressive process. There's an actual set of parameters that emerges that describes the mean syllable. It also describes the distribution of variance around that mean trajectory, right? And so we can look at, in any instance of the data, how well it fits that mean and that variance. And we can ask, are those means and variances similar across different contexts? And that gives us confidence that it's the same syllable in different animals or in different contexts. So yes, it's not via inspection, it's via math. And I'm not going to show you a lot of that math, so you're welcome to look at the paper, but that's how we do it. Okay, so yes?
So I assume the syllable covers the entire period of the observation? Oh, it's actually there are gaps in between.
Yeah, that's a spectacular question. So one of the assumptions we make, there's two kind of underlying assumptions here. One is that there's an identifiable behavior at any given moment in time. So it covers the entire observation period, it's continuous. And the second is that there's only one behavior happening at a time. And so there are modules, and when we identify behaviors, by definition, we're identifying them in this unitary way. So if you do two things, you can imagine walking or chewing gum or walking and chewing gum. So MostSeq would distinguish those three things. It would identify walking and chewing gum at the same time as being different from walking or chewing gum, right? So one thing at a time, but every frame has a label. Okay? Okay, so I want to get meta for a second, if that's cool with you guys. So I just gave you this whole spiel about how it's super powerful, and we can use it to explore all these differences in these different experimental conditions. I didn't show you any data, but you believe me, right? I mean, it sounds great. But why did I do that? What was the purpose of me telling you all that, other than just to show off? So the purpose was to give you some sense that there's some reason to believe that there's something right about MostSeq. It was a way of arguing that there's something valid about MostSeq, of validating this as a technology. And the standard that I held myself to in that argument was one of utility. I had some goal, which is to distinguish these experimental conditions, and so MostSeq helped me do that. Therefore, ... MostSeq is valid. And so a thing that I would like to argue is that this utilitarian standard, which is the one that collectively behavioral neuroscience has held itself to for 100 years, has led to some problems, right? So one of the basic practical problems it's led to is that we cannot have conversations with each other really about what constitutes a better or worse description of behavior, because by definition, whatever is a useful description of behavior is whatever gets my paper into Cell Science or Nature. My paper got in, so it must be a useful description. And that's literally the level at which we've been having conversations. And so people who are traditional behavioral neuroscientists often argue amongst themselves about what constitutes a good description of behavior, but they're really just arguments with words. They're arguing about semantics. What's clear, right, is that this technology, this technique, MostSeq, is the beginning. The things that I've been working on, Kristen has been working on, and many others have been working on, I think really represents the beginning of a revolution that's taking advantage of advances on the hardware side, in machine vision technologies, and certainly on the computing side and in the machine learning side. And so we're kind of at the very beginning of a place where, in the end, we're going to be able to, in a very high-dimensional way, characterize comprehensively the pattern of behavior of any animal in any context. And that's being coupled, of course, with technologies that are farther along in development that are ultimately going to allow us to probe the dynamical activity of tens of thousands of neurons in parallel. And the challenge we face as neuroscientists is going to be to relate those two types of data as we're beginning to confront. And that's going to require us, I think, to think much more seriously about characterizing behavior and knowing what we mean when we say we have a better or a worse characterization of behavior. So I'm going to argue, and it's just an argument, that perhaps a practical way to think about that is set aside from a utilitarian argument is prediction. That a description of behavior is better if it lets you make more accurate predictions. Okay? And I'm not going to specify what you should be predicting. I'm agnostic to the what. I'm just going to say that. Right? And so there's many different things you can imagine that a description of behavior could predict. One thing you could imagine predicting, which Kristen talked about, is future behavior. So you can imagine this is a completely enclosed system, and the thing you want to predict is the animal's future. Literally, that is how motion sequencing identifies behavioral syllables in grammar. It tries to maximize its ability to predict future behavior because it is a generative model, because it effectively represents, you can essentially play MostSeq forward and generate a fake mouse, and then ask, how well does that fake mouse match data that comes from a real live mouse? And one of the things we can do, which is exactly what Kristen showed, is take a synthetic walk through this space and generate a fake mouse and do what David Blay would refer to as a posterior predictive check and just ask by eye qualitatively how well the fake mouse matches up with the real mouse. And so that's what we're going to do here. So this is a 3D movie of a mouse. One's real, one's fake. Here we are going to align the mouse virtually along the axis of its spine, so its nose will be here and its tail will be here. And here you're looking at...
Maybe I should interrupt here briefly.
Yeah, please.
We have time, right? And I think this is
yes.
My original, when I gave my talk here, there was a rant about the difference between describing and understanding. And I warned the students about just mapping. This was about neural activity, about behavior. About just mapping neural activity and describing it in great excruciating detail without having a higher level model, like a MAR level 2 model of what this implementation is actually doing. And I got a sense right now from your case that you are sort of driving very hard at the descriptive level, having a really thorough and detailed description of the behavior.
So I disagree with that.
That can actually go against understanding.
Yes,
right? I mean,
I agree with you.
Much more than useful.
Actually, so let's talk explicitly about that. So, like, there are many ways that I could generate a description of behavior and capture its features. And what I've explicitly chosen to do is to do this in a model-based way, in which the components of behavior and the grammar of behavior is kind of, is very explicitly described. And that is a hypothesis, and so you might think carefully about why it is behavior would be organized in that way. And that is and what's important about that is that there's been decades of work and people thinking about how behavior is organized and how that might relate to motor control. And essentially, this description of behavior services that hypothesis. And what I will show you in the second half of this talk is how the fact that we made this choice to describe behavior as being modular allowed us to understand brain activity. So you might imagine that So from our perspective, we actually made many compromises in terms of our ability to predict behavior. You can imagine if we took a strictly deep learning-based approach, I could build a deep learning model that would do extremely well at predicting behavior for many more than just a few seconds, right? But that deep learning model would tell me nothing about the underlying structure of behavior. Here, I'm imposing structural elements on purpose in the hopes, in the hypothesis, that those structural elements will ultimately teach me something about the brain. And I will show you that's at least in part true.
Or in other words, I mean, you can also simply in behavior go up and ask why is the structure of the behavior actually adaptive for the animal?
Right, that's the next level, right? That's
this
purposeful.
You don't have to go to the brain for that. Right?
And that's kind of what I meant, is that you can think about, from a utilitarian perspective, you can think about predicting lots of different things, right? And I'll show you 3 different things that we can predict, one of which is future behavior. Okay, so that's shown here. So one's a real mouse, again, in a line space. One's a fake mouse. And I just want to mention that the real mouse is viewed, in case there's some grumbling about this, is viewed with our first generation camera, which is like, I think 16 times less resolved than our current cameras. And there's lots of smoothing that takes place over the data, so that the animal looks much more like a jelly bean than a mouse. But since we have these higher resolution cameras, we can ask whether or not these higher order spatial features really matter for a lot of what we care about, and it turns out that it generally doesn't. And so this is a pretty accurate representation of what matters about a mouse's pose dynamics, this kind of jelly bean model. Okay, so you have 2 movies, one's real, one's fake. And these are in line space. It takes a little while to get used to these movies, but you can tell that when like a mouse is a circle, it's orthogonal to the camera. When the mouse is kind of rhythmic, it's going to be rhythmic there. It's like running. You can see when the mouse is bent, it's turning to one side or the other. And my question to you guys is, which is real and which is fake?
Right is real, left is fake.
Any other opinions? All right, so as in most things, Florian is correct. The right one's real. I said most things, not all things. The right is real. You can tell because there's some sensor noise here that lives in this imaging that's not here. From a qualitative perspective, this gives you the qualitative sense that our model is pretty reasonable. But the other thing we can do is actually quantitate something called held out likelihood. We can ask we can quantitate explicitly how well, quantitatively, in a relative sense, does one model predict behavior when compared to another model. And what that means is that we can now take apart this ethological hypothesis of behavior and articulate all sorts of other alternative models for how behavior might work and compare them by asking their relative ability to predict future behavior, which is getting at what you were saying. So an alternative hypothesis, a totally reasonable one, is that behavior is perfectly continuous. So you build a model in which you just have an animal, you have a continuous behavioral space, and animals take a kind of continuous trajectory through that space. Another perfectly reasonable model is that there aren't really pose dynamics, but they're just kind of poses the animal adopts that are kind of related to each other, like clusters in a pose space, and then it switches from cluster to cluster, one after the other. And we've built a huge set of families of models that instantiate all of these hypotheses, and then you can, by computing held-out likelihood, effectively the ability of the model to predict future behavior, you can actually bake these off against each other, right? And so, of course, we are using the highest performing model, the AR-HMM, which is, which has this, the AR is the syllable component, and the HRM is, and the sticky.
What do those letters mean?
So this is the model we use. It's an AR semi-Markov model, which describes the model structure, which means that behaviors are built out of modules that switch one after the other. Oh, autoregressive.
Oh, thank you.
It describes how we think about the individual behavioral syllables. You can imagine that that each syllable could instead be a Gaussian mixture model, and we can compare a mixture model which doesn't have dynamics in it to an autoregressive model, and you see that having some dynamics is really helpful. Right? You can imagine that there's no switching at all, that there's just a continuous autoregressive trajectory through post space. You can imagine you just have clusters. And what you can see is that this generative modeling framework allows us to explicitly bake these off and to pick a winner. And that is something that has never been done before, really, in behavioral neuroscience. The idea that because you're modeling behavior generatively, you can begin to bake off models. One thing that I will assure all of you is that this AR-HMM that we're using now as part of MOSEEK is deeply flawed and inaccurate and doesn't capture many behavioral dynamics, and it accurately represents others. And I am certain that many of you in this audience can come up with better models for behavior. And what's great about this framework, and the thing that I'm really arguing, is that we can now, we don't have to argue with words. We can just measure held-out likelihoods and ask which model does better or worse.
I guess the kind of counter-argument to that is we don't necessarily know the right way of measuring how well we're predicting future behavior.
That's correct. Chris and I agree with you, yeah.
If you knew what that was, you would just optimize that.
Yes, exactly.
That would be the best outcome.
Another way to say this, right? Just a different way to say this is that this is only a relative prediction. We have no way of really knowing the best way of knowing how close to the truth you are, right? We don't like this. This actual number could be much higher than this.
I think it depends. Like if you have one way of saying how well you're predicting future behavior. Right? You have some Euclidean distance in some space.
Yes.
Or something like that. But that space might not be the right space to be looking at. Euclidean distance might not be the right space to look at. And
this is assumption.
The thing that kind of always happens with machine learning approaches is if you come up with the criterion of what you care about, then you just directly optimize that criterion.
Yeah, I think the only thing that I'm arguing about is that we should.
Yeah, I'm not saying it's a bad thing to do.
I think it's more than what we've done. Yes, yeah. I think I'm arguing for the existence of rulers, right? I'm not arguing for necessarily whether we should be measuring feet or meters.
But
since we're in Europe.
But we don't know what would be perfect. Is that the?
Yeah, I think that's the summary of that.
But couldn't you simply? I mean, if you have 80 syllables.
Yeah, so that's
the perfect sequence, right?
That's kind of what Kristen's saying, though.
The mouse will do 22, 14, 16, 87, and if it does it, I'm 100% correct, and if it doesn't, then I.
So you can, so that is the thing you can do in terms of predicting labels. But that assumes the labels, right? That's the thing. It assumes the labels, right?
What it does though is like if number 22 and 23 are closer together.
Exactly.
In some way than
That's right.
22 and 24, it doesn't incorporate that, which you might actually care.
Yeah, that's exactly right. So again, I think this is a way to think about things. I think it really argues strongly for a generative framework. That being said, there are assumptions that are built into this kind of comparison. And again, I'm not going to argue for a particular way of predicting, just that predictions are useful and a way we should start thinking about this as a problem, right?
I think the solution, or a solution, is to have lots of ways of measuring predictions, and some algorithms will be better at lots of them.
Absolutely, yeah. And actually, I think that I will just reiterate here is that these predictions are context specific, right? Like in this case, I'm making predictions based upon a particular set of training data and a particular set of held out data, which are about particular mice in a particular context. And actually, the way these models perform and the methods that you use to characterize behavior will vary depending on context. So it's definitely not it's definitely possible that a continuous description of behavior could apply in a particular context. It's just in the open field with mice, it seems to perform less well in this particular context. Is that fair enough? Okay, so here we're talking about predicting future behavior. There's another kind of prediction that you can make, which is exactly the sort of prediction that Kristen was talking about before, which is that you might actually want to predict different experimental conditions or experimental manipulations. And that's an important thing to think about because, for the most part, people aren't building generative models for behavior. Traditionally, that hasn't been done. We have lots of different ways of more conventionally describing behavior, including kind of point tracking methods, which we've talked a little bit about, which are much more widely adopted. And it would be nice to be able to compare those point tracking methods or other methods, more traditional methods, to these kind of newer methods that are being developed now. And one way to do that, to kind of level the playing field, is to ask how well each one of these methods does at discriminating between different experimental conditions in an experiment. And so we decided to build a kind of reference data set that would allow us to, in a very general way, do these kinds of comparisons. So we took 1,000 mice. We treated each mouse with one of 15 different neuropsychoactive drugs, which ligate a wide variety of neuromodulator systems and neurotransmitter systems, thereby causing a huge diversity of behaviors. We did this at 2 to 5 separate doses, so it's a huge experiment. And we then took each of these animals, acutely threw them in the open field, filmed them for 30 minutes, and then from those data built either kind of a traditional behavioral fingerprint for the mouse, where we did a bunch of point tracking and we measured a whole huge number of parameters, which I'll describe on the next slide, or we did motion sequencing. Then we just took those data, we fed them to a simple linear classifier, and we just asked which type of analysis affords us more insight into what drug and dose each animal got. Okay, so I just want to show you these fingerprints because I think they're cool, and they have a lot of explanatory power, actually. So this is so on the y-axis are all the drugs we tested, and color-coded are you know, a high and a low dose for each one of these, although we did many doses for many of these drugs. And here I'm just going to depict a subset of the traditional things that we measured about behavior that went into those traditional behavioral fingerprints. So we included the complete distribution of positions the animal adopted in the arena, all of its speeds, all of its lengths, and data that is not traditionally available with like a NOLDA system, but we have all of its heights. Okay? And there's actually many other parameters that went into this: the ellipticity of the mouse, its heading, its direction, you know, like its head orientation, distance between the head and the tail. You can just invent a whole bunch of these and concatenate them into a giant kind of description of behavior. And what's nice about this particular data description, I think, is that it explains why we describe behavior using these kinds of terms really nicely. So if you look at high-dose Haldol, which is shown here, you see that the mouse's position becomes fixed in the arena and its speed goes to 0, and that's because in mice, high-dose Haldol does what it does in people, which is to knock you out, and you can just see that with your eye right here in the data. You can do the kind of the converse experiment. If we give high-dose meth, of course, the position goes to the edge of the arena and the speed goes to infinity because the mice are spinning, right? They're running as fast as they can because they're high, just like humans on high-dose meth. So that all makes sense, right? For motion sequencing, we decided to handicap it as much as possible and included only 0th order statistics about behavior. So we include just in each behavioral fingerprint how often each syllable was used. And we turn that into a barcode. And this barcode does not give you any insight in the way that these kind of traditional measures do. This is just kind of nonsense. But if we feed these 2 data streams to a linear classifier, we learn something. So here this is a confusion matrix. On the y-axis are all the different drugs we tested. On the x-axis are all the drugs our classifier on a mouse-for-mouse basis thought the animal was exposed to. And you can see that if we just kind of have these traditional behavioral metrics, you learn very little about the name of the drug that any animal was exposed to based upon its behavior. But with our current models, 97% of the time, MostSeq can tell you the name of the drug the animal was exposed to based on behavior alone, and 93% of the time it can tell you the drug and the dose. That means distinguishing between, like, acutely between 2 slightly different doses of an antidepressant, for example, based upon a behavior that's being expressed in a featureless circular open field. And so this is essentially quantitative evidence that from this perspective, from the perspective of this classification task, MostSeq significantly outperforms kind of just point tracking that you would get, say, out of Deep Lab Cut or LEAP using current methods. And so this is very useful for us because it provides a kind of quantitative basis for making the claim that MostSeq has some sort of predictive power that exceeds that of other metrics. Now, what's nice about this data set that we built is that we can now begin to compare lots of different methods and ways of thinking about behavior. So one thing you might argue, which I've argued, is that this comparison is unfair. And it's unfair because what I'm doing here is just describing the mouse's behavior as kind of a histogram, a distribution of a bunch of different behavioral parameters. There's some sort of grab bag of information. And there's nothing about time or evolution of behavior at all. There's just some sort of overall bulk description of behavior. And so one might want to know, if I feed these data to a time series model of behavior, say like MostSeq, do things get better? Like, if I just take tradition, if I get rid of the 3D video and instead just take point tracking data and feed it to this classification program, how do things do? And so we can ask that directly. We can actually summarize, I'll just go back one slide. We can summarize the on and off diagonal content of these graphs by computing something called an F1 metric, which are these numbers here. And you can see, as I showed you before, motion sequencing significantly outperforms just scalar metrics. But if I feed these scalar metrics to MoSeq, I definitely do get an improvement in performance, which is the thing you'd predict. You predict that behavior is a thing that happens over time, and so using models that respect time would be useful and would improve your performance. But it doesn't actually get you near the performance of motion sequencing. And so from a quantitative perspective, we can use this kind of data set to begin to bake off different ideas about how we segment out behavior.
Could you use the information you have now to specifically tailor your analysis to extract something?
To maximize performance here. So yes, we've begun to do something. We've begun to do a little bit of that to try to maximize performance because now we have a different thing that we can predict. Of course, that's something that Kristen does as well. I think it's a very useful way to think about taking advantage of this kind of data set. So the other thing we can do, right, is not just bake off kind of more traditional ways of thinking about behavior, but bake off more modern ways of thinking about behavior. So I think a very useful method and a very powerful method and generalizable method that has recently been developed has been developed by Gordon Berman. It's called Motion Mapper. And it's a technique in which you feed it videos, and it's very general. You can feed it videos of flies or humans or mice. And it essentially does a kind of wavelet transform, embeds those data into a two-dimensional space, and then performs clustering and describes behavior through these clusters. And so we can essentially generate a motion mapper description of our data set and compare that to motion sequencing, effectively comparing clustering to a kind of more dynamical time series description of behavior. And again, we see that motion sequencing outperforms motion mapper, suggesting again that behavior is a thing that happens over time, and that having models that our time series models will outperform methods in which you just cluster behavior in various ways. And so I'm going to end this kind of prediction part of the talk. I'm going to end this part of the bake-off part of this talk. But I just want to reiterate that, again, like the claims that I'm making are context-specific. Motion mapper is very general in a way that motion sequencing is not. And so you can easily imagine situations in which it's a better tool to use. Clearly, tools like Deep LabCut and Leap and Java are very, very generalizable and allow individuals to take any sorts of videos from all sorts of cameras in the wild or in the lab and do effective tracking of behavioral features, and that's something that definitely motion sequencing does not allow you to do. So I'm not claiming that motion sequencing is the solution. It is a solution. I'm just arguing that we should think hard in a given context about how we compare different methods, and one way we can do that is through this particular kind of prediction. So the last kind of prediction you'd want to make, and the one that I want to talk about at the end of this talk, is to predict neural activity from behavior. So of course, as Florian and I were talking about, the reason why we like this ethological model is essentially because we believe the ethological hypothesis. And so that predicts a very specific thing, which is that somewhere in the brain there should be some neural correlate for these syllables or for the grammar. So the question is, how do we go find those? Because the brain's a big place. Right, so our thinking was guided by this experiment. It's a very simple experiment. Now we're not in a circular open field, but instead we're going to be in a square odor box. So each one of these corners actually houses an olfactometer delivering odors into this box. We're going to look again at 30 separate animals from 30 separate experiments. We're going to overlay those all into a master movie. And I'm going to outline the position of each mouse in this experiment here, and I'll show the average position across the whole experiment of all the mice in gray. And at the beginning, I'm just going to blow air through all these 4 ports. So what you're looking at is normal open field behavior, and as Andreas showed you before, mice love these corners, so they tend to congregate in these corners and to explore them. They don't like the center so much, but they'll dart through the center occasionally, as you can see. That's normal behavior. And now we're going to blow this odorant that Dayu talked to you a little bit about, called TMT, which is an aversive odorant from fox, which is thought to evoke fear-like responses in mice. And you can see that indeed mice avoid this TMT. They do this kind of darting behavior and sampling behavior where they come into the odor port, they smell it, and they run away. And they tend to actually physically freeze in these corners.
There's always a single mouse in there.
For any experiment, there's always a single mouse. There's some interesting new things you learn if you put multiple mice, but I'm not talking about that. This is just overlaid for convenience. So the question is, from the perspective of motion sequencing, what did the odorant do to change the animal's apparent pattern of behavior? So we can compute these behavioral state maps, and if you look at these behavioral state maps very carefully, you'll see that each one of the behavioral syllables that's present in this map is also present in that map, although they're expressed differentially. And so that tells you that a kind of surprising thing, which is that the new behaviors that are induced by TMT, those new behaviors don't require the expression of new behavioral syllables. Although our system clearly can identify new behavioral syllables, it's not in this case. Instead, what's clearly happening is that the transition statistics that govern how different syllables are interconnected are being changed as a consequence of exposing the animal to this ethologically relevant odorant. And you can really see that if you subtract these maps. So in blue are all the new trajectories through this behavioral state space that are upregulated, and in red are all the trajectories that are downregulated.
Just for understanding, what is the position of the syllables within the big circles?
Here it means nothing.
But when they're close together, they're more similar to each other? Here
it's just a way of embedding the data. It is not true that there's... And actually we've actually...
But the size is how often...
Can I just mention something about that, which is actually really important? Yeah. So one trivial thing you might imagine being true that would tell us that we had messed up our behavioral segmentation is if the things that are near each other always were like very, very similar things. Right, because then it would be like, actually those are really the same thing, and they're interconnected, but that's because you're not segmenting behavior correctly. That's not true. So we've looked at. So here, it's a variant of a force field-based way of embedding the data just to organize it.
But the size tells you something about the relative frequency.
Yes. The size tells you how often it occurs, and here we're looking at differences, TMT-induced differences in how things transition. So everything that's new is happening here in this blue part of the space. Everything that's not new, but like all the new behaviors must be encoded via trajectories through the space. And again, because motion sequencing is transparent, that is, we're not just like clustering data in some high-dimensional space. We're generating videos of what each syllable is. We can actually just look at trajectories through this space and ask whether they encode the new behaviors that are induced by TMT. So now when I show you a dot, the dot encodes 3 behavioral syllables in sequence that are all blue, as opposed to before where it was just 1. And you can see that indeed these blue trajectories encode the freezing behavior that the animal was generating before, that the animal generates as a consequence of being exposed to TMT, and this kind of motivated odor-guided avoidance of the specific quadrant in which the TMT is being presented. And so this tells you something really important. It tells you that adaptive behaviors aren't just kind of de novo patterns of pose dynamics, but instead the brain somewhere within it contains some sort of machine that can resequence behavioral components to create new patterns of action. So the question is, where would such a machine exist in the brain? And that got us, of course, thinking about the basal ganglia, and in particular, the input nucleus to the basal ganglia, a part of the brain called the stratum. And that's because, of course, many decades of work have identified the stratum as a key node in motor control, and in particular in 2 things that are relevant to our problem here. The first is in motor implementation, that is the ongoing execution of actions. And the second is action selection, the process through which animals decide at any given moment in time to express one behavior at the expense of all others. Now, I'm not going to give you a big long lecture. Go ahead, Tayu.
Yes, so just before we move on to the neurosubstrates of the syllables, I just want to get a little bit further sense about how stereotypical they are. So like, under those drug conditions, did you find any new behavior syllables?
Yes, of course, yeah. So yes, absolutely. So I can tell you more about the drug. The drug experiment turns out to be very interesting. So there's some drugs, for example, like high-dose amphetamine, that induce stereotypies. So these are well-known stereotypical, like repetitive behaviors that are OCD-like. Those are not behaviors that are normally expressed by mice, and those are nicely encapsulated by MoSeq as new behavioral syllables that are unique, say, to high-dose amphetamine, right? There are other changes in behavior that are much more subtle, that involve just the modulation of how often a particular behavioral syllable is expressed. So here's just kind of a thought experiment, and there's an interesting answer. So you might imagine that if you kind of use drugs, all of which are nonspecific, by the way, right, to kind of manipulate behavior, that there's one of 2 possible ways in which motion sequencing reveals how these behaviors are different. So one way is that each drug could affect every behavioral syllable. Right? There's some exceptions, there's some new syllables that aren't present in others, but maybe that just in general, drugs diffusely affect the usage of behavioral syllables. So you can imagine each syllable is a key in a piano. You can imagine this is the slamming your arms down on the piano keys model. The other possibility, right, is that drugs actually very specifically influence subsets of syllables, and those subsets then are useful biomarkers that allow you to identify when a particular drug is in the animal system. And it turns out that it's the latter thing. So individual drugs and individual doses modulate the expression of small numbers of behavioral syllables that are kind of like chords, and those chords are characteristic of each drug. And that turns out to be really, really useful, because you can imagine characterizing genetic models of disease that you care about, and characterizing how syllables are changed in those models of disease. And it turns out in most of the models that we characterize, We get chord-like changes in behavior. So we get changes in limited subsets of behavioral syllables. So for example, in autism strains, typically we get changes in subsets of 10 behavioral syllables or less or fewer that characterize the main behavioral change in that model. And since drugs also give you a chord, you can ask how those chords intersect. And that allows you to know specifically about the on and off target effects of drugs in mouse models of disease. So a whole kind of thing that we're doing on the side in our lab is taking advantage of this chord-like nature of behavioral changes that is revealed by motion sequencing to get a handle on how drugs and genes interact.
I'm curious, how do you deal if the MoSeq identifies syllables which are actually, that you judge as similar? Let's say, if we talk about syllable turn right? I guess the MoSeq will give you 10 different, or I don't know about numbers, but several different turn rights.
Yes, that's correct.
Yeah, and apparently it does not, it basically separates them. It does not say that it is similar, right?
It separates them based upon their ability based upon the idea that the separation of those turns allows the model in a generative mode to better predict future behavior. That's why, so it doesn't just arbitrarily separate them. It separates them it agglomerates them or separates them based upon whether or not you make better, it allows the animal to make, the model to make better predictions. So just to be clear about that, so there are many different forms of rearing that emerge. There are many different forms of turning. If we look at the videos, I've actually, like my graduate students who work on this project hate me because I always insist that they actually look at each of the videos and actually give labels to each of them by hand so that we can talk about them, right? And they, after looking, they can see the differences, right? So the model spits out idealized trajectories and noise distributions, that characterize each syllable, right? And so we can know mathematically that those are different. And when when humans look at the data, they seem different, although they're clearly related. And they can be, and I'll show you, just a few slides, we can actually organize them hierarchically. So we can understand the relationships between these, we can understand, and we can group them. And that grouping can also be very useful.
Do you also see the sequences or syllables that for a human look similar or the same, exactly, but
it's exactly, what we see is exactly what ethologists see, which is that if you just look at a couple syllables for a second, they look the same to you and you can't tell them apart. But if you get more data and you make more observations, then the differences become very clear. So like these differences are often very confusing for a naive observer, right? And they become very obvious to someone who's seen a lot of data. And that's exactly what ethology is, right? Ethologists get better at segmenting behavior the more observations they make. Right? And that's because their certainty over differences and similarities in the data grows as the amount of data they have increases, right? Okay, so to the stratum. So the stratum is largely composed of inhibitory medium spiny neurons, as you mentioned yesterday, and these come in two flavors. About half of them express the dopamine D1 receptor, and these neurons comprise the so-called direct pathway, which is thought in some way to facilitate action. The other half express the dopamine D2 receptor, and these neurons are thought in some way to inhibit action. But how these two pathways actually interact during naturalistic behaviors to support the generation of meaningful behavioral sequences is not really known. There are two kind of cartoon extreme models that are useful in our thinking. One emerges from work of Anatol Kreitzer and others, in which these two pathways are fundamentally competitive, and that predicts that if you were to record from these two pathways, they should be anti-correlated, that one pathway's on, the other's off, and vice versa. There are other models from folks like Rui Costa that suggest that these pathways collaborate in an ongoing way to help select actions, and that predicts the opposite, that these pathways should both be on as animals generate behavior. But I think we definitely don't understand how these pathways are mechanistically interacting to give us coherent patterns of action. What we do know, go ahead.
Who's right?
I have we'll talk about it.
But that should be a relatively straightforward experiment.
It's not straightforward because it's been very difficult to do two things. It's been very difficult to simultaneously record from both of these pathways. Okay, so that's one technical challenge, and it's one that I'm going to address. And the second thing is that part of your thinking about what is correlated or anti-correlated requires timestamps for behavior, and people's timestamps for behavior have really been bad. And so I'm going to talk about that. That's exactly what I'm implying, is that there's this kind of controversy that has a technical basis, and we can think about it if we improve the technology. Okay, so the one thing that is definitely true, right, is that if you stick an electrode into the stratum, you recover lots of information about behavior, right? So if I go ahead and do that and I let a mouse or a rat run around, there are neurons that encode the animal's velocity, the direction of its head. If I do this in a monkey, as the monkey grasps an object, there are clearly neural correlates that encode the specific kinematics of this grasping behavior, and that's consistent with this first idea that the dorsal striatum is involved in action implementation and generating movements in a coherent way. But it's clear that there are also neural correlates that exist in striatum that encode much more abstract structural components of behavior, like information about behavioral components and behavioral sequences. And we know this from learned behaviors, from important work from Rui Costa and Graybiel, and we know this also for naturalistic behaviors from important work from Wayne Aldrich and Kent Berridge. And in the 80s and 90s, they did this incredibly painful experiment, which is to film rats with high-speed video as they just were kind of hanging out in an open field. And then they did frame by eye classification of the animal's behaviors. And what they discovered was that when animals groom, they tend to groom in a particular sequence, and they could essentially identify components of that grooming sequence. And when they went ahead and did neural recordings specifically in dorsolateral striatum, they could identify neural correlates, neurons that fired during specific components of a grooming sequence, but these correlates only fire if the grooming sequence is being completely expressed. And that suggests that, of course, in addition to encoding information about the ongoing expression of movements, that the striatum during naturalistic behaviors is also encoding some information about that's relevant to the structure of behavior, the components and the grammar of behavior. And so a postdoc of mine who's joined with Bernardo Sabatini named Jeff Markowitz, along with a student of mine named Wynne Gillis, were really inspired by this work. And they hypothesized that maybe the dorsolateral striatum in general contains information about naturalistic behaviors, not just about grooming, but maybe there are actually neural correlates in the dorsolateral striatum for syllables or grammar. And in addition, they were interested in this question, which is, given that we have this nice behavioral segmentation and the ability to call behaviors out over time, how are the direct and the indirect pathways interacting? So they did two things. One is they rendered motion sequencing compatible with all forms of neural recording with tethers. So now we can do motion sequencing, and that required a lot of machine vision stuff I'm not going to talk to you about. But now we can do neural recordings while we're doing ongoing silicon probe recordings to do electrophysiology. As I'll show you, we can do Inscopics type recordings, and we can do fiber photometry. The second thing they did was to develop a new multicolor fiber photometry system that allows us simultaneously to assess bulk activity in the direct pathway and the indirect pathway simultaneously as we perform motion sequencing. So if we go ahead and do that, we put all of this together, this is what we get. So this is a 3D movie of a mouse. Again, it's in a line space, so its nose, it's freely exploring the circular open field, but its nose is always going to be pointing to the right, and its tail is always going to be to the left, so you can see its behavior. But you see this animal is now wearing a hat. Out of the hat is coming a fiber. That's because we're performing dual color fiber photometry, specifically in the indirect and direct pathways in the dorsolateral striatum. So this movie is being compressed down into this movie, and I want to show you this movie, which is just the raw pixel data, because when you look at this movie, you see that it's very striated. Okay? And that's where we got our prior, that behavior is organized at the sub-second time scale. Because if you just do standard kind of frequency analysis over these data, it's quite clear that there's a kind of switchiness to behavior that's just intrinsic to the raw data that we can't get rid of, and that's visible by eye. And that's where that prior over the model came from. Below it, we're going to use MoSeq to, on an ongoing basis, call these syllables. And down here, we're performing simultaneous direct and indirect pathway measurements. Direct is D1, indirect is D2. And you can see that sometimes these are highly decorrelated. Other times, they're highly correlated, right? And so what this means is that one model is not right and the other model is not right, but there's some complicated interaction between these pathways that governs how they're influencing behavior. So one thing we noticed just in general as we looked at a lot of this data by eye is that as animals switched from one syllable to the next, we tended to see a little blip in activity in one or both pathways. So we just wanted to ask whether that little blip was a true thing. And so we averaged all of the data. So on the x-axis here is the time switching from any given syllable to the next syllable, where time 0 is the switch. And here we're just going to look at activity in both pathways. And what you see is that systematically, on average, as animals switch from one behavioral syllable to the next, we get this fluctuation in neural activity that's apparent on average in both pathways. We did some simultaneous unit electrophysiology along with this multicolor photometry to actually calibrate the photometry to know how electrical activity actually is reflected by these photometric signals. And what that taught us was a simple thing, which is that if we took the derivative of the signal, we could get a pretty good sense of the onset at which electrical activity was occurring. And that teaches us that about 50 milliseconds before an animal switches from one behavioral syllable to the next, you get this blip in activity in the striatum. And so for us, this result was really, really conceptually important. As we've been talking about, we use motion sequencing to segment behavior, and it segments behavior based upon the structure of behavior alone, with no other information, right? And so that raises questions about how valid that segmentation is. Here we've used that segmentation to effectively discover a new hyper-ultradian rhythm in the striatum, which I think provides a kind of important biological validation that something about motion sequencing is useful or real. And that's exactly the sort of thing that we were talking about before. Okay, so one question you might ask immediately, given these data, is does each behavioral syllable result have some high level of activity in both of these pathways then declines, or alternatively, is each behavioral syllable kind of afforded its own unique amount of activity in these two pathways? And so what we can do is a very simple thing, which is just to average the neural activity in the direct and the indirect pathways during the expression of any syllable, and then plot that out. So you had a question?
Did you do that when the animals were doing the grooming change? Because I remember that came very challenging. When they recorded from the stratum, the activity was different if the same behavior was being done.
I'm absolutely going to get that. I'm going to get to that. I think it's a critical thing, and I'm going to talk about it explicitly. Okay. Okay. So is this true for all syllables, or is this just what we see on average? So if we go ahead and plot out activity in the direct and the indirect pathways, we're just going to color code each behavioral syllable, and this is across like 12 different animals. What you see is that each behavioral syllable correlates with a unique amount of average activity in the direct and the indirect pathways. And because these are color-coded, we can know that across individuals, these patterns seem to be conserved from animal to animal to animal. That is, this behavioral syllable in this individual has high levels of activity in the direct and the indirect pathway, and that's true across individuals. Okay?
And that's true just during the transition?
This is true during the expression of the syllable. Yeah, exactly. So like during the expression of the syllable, what do we see in the stratum? So we see that on average, there is some kind of constant relationship between the amount of activity you see and the specific three-dimensional pose dynamics that are observed. That suggests that the stratum contains a code, a neural code for the animal's three-dimensional pose dynamics, right? That's the first thing. The second thing I want you to notice is that on average, the activity of the direct and the indirect pathway here is highly correlated on average during the expression of each syllable. And that's consistent with this Rui-Costa idea that these pathways are being simultaneously activated as part of the process of action selection. Okay, so I just want to point that out. Okay, so this is what we see again on average, but of course we have these beautiful time-resolved waveforms that are associated with each behavioral syllable. What do those waveforms look like? Do those waveforms convey any information about the behavior the animal is generating? So we can just take 3 syllables at random and plot out the waveforms that are associated with each one of these syllables. And here, of course, these syllables are each pretty brief, right? On average, 300 milliseconds long. So we have tons of data. Here we're averaging thousands of different instances of this particular behavioral syllable, lots and lots of data. And what we see is that when we do this, first off, you learn 2 things, right? One is that each syllable seems to be associated with a particular pattern of activity in these 2 pathways. But the second thing you notice is that now that we have this kind of fine time alignment of behavior, we know exactly when a syllable begins, and it's kind of very clearly defined. Now we see these decorrelations in activity in the D1 and D2 pathways that's not apparent when you just average the data. Okay, so there's these fine timescale decorrelations in the activity of these 2 pathways. It's kind of missed if you average out all the data, or alternatively, if you have a kind of sloppy definition of when a behavior starts or ends. Andreas?
Do these fingerprints look the same for the same syllables in different contexts?
Yes, in different contexts. We have done that experiment in 2 ways. We have changed the physical configuration of the environment the animal's in, and we have given the animal TMT, right? I'll talk to you more about those data. And in those examples, syllables have the same neural representations across these different contexts. So that's not all contexts, but it is consistent with this argument that there's a kind of conserved code, and that code isn't moved by context, but the use of the code is moved by context. But that's a really important question, and one that we directly ask, because yeah, we're curious whether or not every time the animal moves into a different context, the code changes, right? That'd be a big problem for the way that we think. Okay, so this is what. How does the code compare? Yeah, also conserved, right? Also conserved, exactly. And we do. I'll show you that we can do some. I'll show you. Okay, so these are 3 example syllables. But one thing again that you notice is that each syllable, which is morphologically different, has a different code associated with it. So the question is whether or not this relationship is systematic. That is, do similar behaviors are they associated with similar patterns of neural activity? So is there some systematic representation of this 3D behavioral space? So in order to address that, we have to somehow think about the relationships between these different behavioral syllables. And so we kind of built out a way that allows us to hierarchically organize relationships between behavioral syllables. I'm not going to explain how that works, but we can actually use this kind of hierarchical embedding to identify different categories of behaviors and to ask and to mathematically define whether two syllables are similar or different. And so we can draw two walking syllables, two scrunching syllables, and two rearing syllables, and now compare them. And what you see is that similar behaviors tend to have similar representations, but that different types of behaviors tend to have different representations. Again, consistent with this idea, there's a systematic representation for 3D pose dynamics in dorsolateral stratum, an intuition that we can formalize by just computing the correlation between neural distance and behavioral distance. And what you see is that if we do that using just the direct or the indirect pathway data, we get some correlation, but it's pretty poor. But if we take advantage of both pathways, now we get quite a nice correlation. And what that means is that the decorrelated activity of the direct and the indirect pathways is usefully contributing to the encoding of 3D pose dynamics in the stratum. Now again, these waveforms themselves are averages, again, more averaging, right? They're the average from Bernardo Sabatini's computations, probably of about 2,000 medium spiny neurons are contributing to each one of these little waveforms. And what we really care about are the neural ensembles that underlie these waveforms. And so as I mentioned, we can do inscopics recordings in the context of motion sequencing. So here we're going to do that. Here the hat has a inscopics camera attached to it. And we can identify syllables, and we can identify activity in individual spiny projection neurons and watch how those correlate with the expression of individual behavioral syllables. And even crudely here, in this little snippet, you can see that as animals transition from one syllable to the next, there tends to be this kind of burst of activity that's associated with the expression of any given behavioral syllable. So if we treat these data like as photometry data and just average it, we again see this systematic relationship during behavioral switching, where we tend to get these bursts. And if we do the kind of correlation analysis I just showed you, we ask about correlations between neural ensembles and the different behaviors the animal expresses. Again, we see some correlations in the indirect pathway, in the direct pathway, but we see the best correlations between brain and behavior if we consider both pathways. One thing these ensembles let us do, of course, is actually attempt to decode behavior. Can we use the specific neurons that are firing at any given moment in time to predict which specific behavioral syllable the animal is expressing at any moment in time? And so we can build decoders, as you've heard about already at this meeting. And what you can see here is that if we take a bunch of spiny projection neuron data from DLS, and we have a behavioral state map that has 41 separate behavioral syllables in it, so it's a ton of behavioral states, very complicated description of behavior, 25% of the time, we can tell you exactly which syllable you're expressing based upon neural data alone. And if we relax that a little bit, if we look at nearest neighbor syllables, so you have to predict either the syllable you're expressing or if you just merge those syllables effectively, this number jumps to closer to 80%. So we can make highly accurate predictions of which behavioral syllable you're expressing at any given time based upon looking at just a few hundred neurons in stratum. This plot is a little bit complicated, so I'm not going to explain it. I'm going to summarize it. What it reveals is that based upon the particular pattern of firing a given neuron generates during a particular behavioral syllable, 100% of the time we can tell you whether it belongs to the indirect or the direct pathway. It's also consistent with the idea that those pathways are differentiable and convey different information. Collectively, these results all suggest a role for dorsolateral stratum in the ongoing implementation of behavior. You have the systematic map of three-dimensional pose dynamics that lives in the stratum, so that really suggests that maybe the stratum's involved in executing these behavioral syllables. But what about grammar? We were talking about before. What about the sequencing of these different behavioral syllables? Can we get any information about sequences from the stratum? So we thought about that in a very simple way, a very simplistic way. So imagine you have a series of 3 syllables, A, B, and C, okay? Now focus your attention on syllable B. Imagine that sequence A, B, C is high probability. And then imagine there's an alternative sequence, D, B, E, and that sequence is low probability. Okay, so B is in different contexts, but morphologically, mathematically, B is exactly the same in both the high and the low probability situation. You might expect if there was some sequence dependence, some context dependence to the neural code for syllable B, that the representation for syllable B should be different during expression of high and low probability sequences. So if we plot that out, actually we find that they're very different. So here, if you just pay attention to this little bit here, what you see is that high probability sequences tend to be associated with a little bit less activity in both the direct and the indirect pathways, whereas low probability sequences tend to be associated with a little bit more activity. And this difference is big enough that based upon the specific type, the specific amplitude of activity that we observe in photometry during the expression of a syllable, 80% of the time or so I can tell you whether it's being expressed as part of a high or a low probability sequence. And this very much argues that the stratum's not just encoding information about the ongoing generation of behaviors, but it's actually encoding information about the order in which different behavioral syllables is occurring. So we wanted to ask, which is the more important thing? Yeah, please, Andres.
These are population activities that you're looking at.
In this example, yes, but if we look at cells in Scopics, we see exactly the same thing. And we discovered that cells are bidirectionally modulated, so that some cells are dialed in some sequences and some cells are dialed out. But in the net, the number of cells falls during high probability sequences, and the number of cells rises in the net in low probability sequences, which explains this difference, this difference in the average amount of activity you see.
There could also be smaller, highly active ensembles emerging in the high probability case, no? I mean,
yeah, but we know that answer.
Activity patterns would get more specific.
Yeah, so we don't, I don't want to, that result is not obviously true. I'm not going to say it's false, but the ensembles look pretty similar in terms of like the amount of firing afforded to any individual neuron. That's on average though, so it could be that if we look more carefully, you could be right. The most striking result is that things are dialed in or dialed out, and the size of the ensembles is changing. But it could be that there's neuron-specific amplitude changes that I haven't looked carefully enough at. But thank you for suggesting, I'll look at it.
I'm not sure I'm understanding this exactly, but could it be that B is actually two different behaviors?
Yeah, so what we did was, I think that's an essential question, right? Yes, that's right. So what motion sequencing does, right, is it gives us this mean and this distribution, and so we can ask in these two different contexts, what is the mean and the distribution in the two different contexts? How similar or different are they? So we find they are statistically completely indistinguishable. We can ask about all sorts of allocentric things, right? Like what's the velocity? What's the, like we can just extract other information. All of those things are all identical as well. We were very, that's the main complaint about this experiment, right? So we did everything we possibly could to address it. And I will say that like the ability of motion sequencing to give you this mean trajectory in the distribution is really powerful, right? Because then you can ask exactly this question. Across conditions, are those things really the same thing or not? And here they really are, right? Okay?
In this case, the main differences seem to be before zero, which is before the occurrence of this syllable we actually compared.
There are differences that are both before and
yes,
after. Yeah, but they're different syllables. But there are different syllables in those sequences, so that's not surprising.
Yeah, but it's
because they're two different sequences.
It captures that prior differences, probably more so than the current differences, which is still somewhat qualitatively similar.
No, at zero, at the line, these lines are very different.
At zero, they're very different.
Yeah, that's all that matters.
Mainly contributed by the prior.
Except that what's happening behaviorally at time zero is absolutely identical. So for this whole window, we can't find anything that's different about the animal's behavior. Right?
Yeah, okay.
So you have a stable neural. So in fact, I'd actually maybe argue that what you're doing is making the argument that I'm making, which is that the neural code that's associated with any given syllable is sequence dependent.
It's sequence
right?
dependent,
Yeah.
yes.
So that's fine, yeah.
The starting
Yeah.
point in a way, but also could
Yeah.
that also be a feature, not a feature, let's say a limitation of the GCAM because it's not instantaneous, right? There is a bit of if you record.
Yeah, but again, we performed electrophysiological calibration, and so we actually know how offset we are with respect to the behavior. So it's only 50 milliseconds or so. So we know that. So I'm not actually worried about this being a GCAM artifact.
Well, but the offset of the GCAM itself is a bit slower, so you smear the temporal resolution.
Yeah, that's true. That's true. I don't know. I think that the ability to accurately predict the sequence context deals with that, right? I mean, we can make accurate predictions using these data. So it's not and we can do that merged across many animals, actually. Is there anything else? Yeah, Andres?
Is it probability dependent or sequence dependent? What would you expect to happen if you were to reinforce the unlikely?
This is the last slide I'm trying to get to. Let me get to my last slide. But yes, that's the essential next question. So let's talk about that.
Yes, of course. So
Yeah.
if the representation of a syllable depends on the sequence, do you start using the uniqueness of the representation of the syllable?
Yeah, so it's a really great question, exactly. So these things live in tension. which is why, right, so we can decode, right, so at least we know that with a computational decoder, we can tell the syllables apart, on the one hand. On the other hand, we can decode this as well. So those things definitely are in tension. So there's clearly some space in which different sequence-dependent representations of syllables are similar to each other. Right? But they are definitely, these two things are in tension, which motivates the next experiment, right? Which is to figure out a way to ask whether or not one or the other is more important functionally. And so the way we did that was the dumbest way you can imagine thinking about it, which is that we just used NMDA to lesion the dorsolateral stratum and take it out of the brain. And then we let the animals recover for a couple weeks, and then we performed motion sequencing. And so here you're looking at a sham case on the left and a lesion case on the right. These are 3D movies again. And if you look at these movies by eye, I think we would all agree these animals, the lesion animal looks pretty normal, right? It's behaving, it's blowing the circular open peel, it's not obviously doing something that's weird. If we perform motion sequencing over these two animals, what we find is that the syllables that are expressed are the same between these two models. And Kristen, again, we can do exactly the same thing we talked about just a moment ago, which is we can actually look at each syllable, its trajectory in AR, its AR trajectory, its noise distribution, and they're statistically indistinguishable. So we can do that rigorous test here. So from this perspective, and given this rigorous test, the implementation of each behavioral syllable does not require the dorsolateral stratum, even though the dorsolateral stratum contains the systematic map of three-dimensional pose dynamics. It's not actually required for that. But if we now look at the transitions between behavioral syllables, this is a different type of state map where all the syllables are just on the edge, so you can see the interactions very clearly. You can see that in the sham case, the brain clearly has a kind of set of rules that govern the ways in which different syllables can interact over time. So this is very non-random. It has what information theorists refer to as low entropy. But if we lesion the dorsolateral stratum, all of a sudden this becomes much more random, and syllables begin to interconnect to each other over time much more randomly, right? And this becomes much less organized. And so what this argues is that the main function of the dorsolateral stratum is to sequence these behavioral syllables appropriately. Now, one objection, a reasonable objection to this experiment, is that I just showed you a mouse that was lesioned that looked pretty normal. So who cares about this increase in the animal's behavioral entropy? Because it doesn't seem to mean anything to the animal. I will remind you all of a result that I showed you earlier, which is that when we gave TMT, the way that TMT changed behavior was to impose a change on the animal's behavioral grammar. So what happens if we take these two animals and give them TMT? So on the left is the sham case. You'll see the mouse will approach a TMT source and then run away because it's supposedly a fox and it hates foxes because it doesn't want to get eaten. As mice are, it's kind of an idiot, so it goes back to the source and it's going to sniff it again, it'll run away again because it never learns, just kind of does this over and over again. And so mathematically, this mouse is avoiding the TMT. If we now go ahead and do that same experiment in the lesion case, what we see is that the mouse doesn't treat the TMT like it's anything. It just, even though it's clearly sensing this TMT, we know that, it treats it like it's just another object in the arena, and it doesn't generate this kind of avoidance behavior. And that's a very, I think, I would argue, a pretty powerful result, actually, that suggests the dorsolateral stratum, it really is meaningfully, in a sensory-driven manner, involved in building behavioral sequences that allow animals to adapt to the world. So I'm not going to summarize what I've told you thus far because I want to get to the last couple slides. I just want to mention kind of what we're doing, how we're thinking about things now, and some of the things that we're doing going forward. So of course, the work that I've shown you thus far prompts, I think, a lot of very interesting and general questions. So the first question is, how does sensory information converge on DLS to influence this sequencing? So we're thinking about that from a mechanistic perspective. The second question is, how does the stratum and how do cortical striatal circuits actually build these sequences? How do they actually help coherent sequences of behavior be generated? And of course, we're doing lots of kind of mechanistic experiments to get at that. And the last question is the one that I kind of started with, which is, given a behavioral sequence, how does the animal's ongoing behavior affect sensory codes that then go on to affect these motor codes? And we're obviously actively working on all three of these problems. I want to talk for a moment about the things that we're doing to motion sequencing per se, because as I've mentioned now several times, we view this as an ongoing project. We think this thing that we've built is really just a first pass attempt to better understand behavior. I'm certain that no one will use this technology 5 years from now to be something better or different. And we're thinking about ways in which we ourselves can make this better or different. So the first thing is something that we mentioned, which is that motion sequencing mostly is being used by us in open buckets because that's the simplest context. And I think I just will mention that I think that there's something good about using motion sequencing initially in that context because there's so little information for the mouse in a bucket, the behavioral differences you see, you expect to see no behavioral differences under any experimental condition in such a neutral environment, and yet routinely we recover them. And so from a validation perspective, we thought that that was an important way to start. That being said, there's so many contexts in which this technology would be useful that aren't just an open bucket, and so we've been building different kinds of technologies that will help us with that. So the first is we now are beginning to be able to effectively segment objects out of the arena and identify mice and do motion sequencing when there's like stuff in the thing. We've done some novel object recognition through these kinds of technologies. The second is that we're getting set up to do much longer term recordings. As I mentioned, we typically record mice for a half hour, an hour. And so a typical experiment like that drug experiment is sort of petabyte scale. We want to be able to do multi-week monitoring of individual animals, so that's really data at scale, right? There's a lot of data. We have to think about ways we're going to handle. That's a computational problem, so we're thinking about how to handle that. Ultimately, we want to work towards getting to home cages where we just do continuous monitoring. But we've been working pretty hard at broadening the context in which we can do motion sequencing. We're starting to think about the use of motion sequencing not just in single animals, but in multiple animals, and to think about their interactions. One thing that we've been working on with Lisa is to work on something that we're calling VOSeq, which is to take ultrasonic vocalization data and to pass that through the motion sequencing algorithm, because again, motion sequencing is agnostic to the data that is fed it. We've done some first pass experiments to suggest that VOSeq really works nicely to characterize structure in mouse ultrasonic vocalizations in an unsupervised way. And we're hoping to combine that with some other technology that we're building that allows us to do two animals at once using motion sequencing to build a MoVoSeq that allows us to really understand social interactions. And I'll just mention my interest in social interactions, which I think is in some ways different from those of many of you who have been working for many years on social interactions, which is the thing that I really want to understand is whether I can find a mathematical framework that allows me to know when the behavior of two mice are coupled or decoupled. So for me, I really want to understand whether this notion of body language as a communicative form is real in mice, and whether mice can communicate using their body language at a distance. And we think that motion sequencing is going to be probably useful for that. We've also been actually changing the model class. I'm not going to explain why we're doing that, but we're incorporating some deep learning components into motion sequencing and actually changing the HMM component of it as well. We actually have a paper that was in NIPS a couple of years ago that describes some of this work. If you're really interested in the machine learning components, I urge you to read that. And the last thing I want to mention is getting at something that Andreas was mentioning, which is that we've finally built out techniques that let us go closed loop over MoSeq. That is, on an ongoing basis, in real time, as animals behave, we can know which syllable is being expressed. That allows us to trigger optogenetic stimuli on essentially a moment-to-moment basis in response to the expression of particular syllables. And so we've done that by generating a lot of training data. We have a lot of video in which we've called syllables out via MoSeq. Again, this has to happen offline in the cloud, but what we can measure in real time is, of course, very simple things like the three-dimensional velocity of the animal or its length in pixels. We can measure that in microseconds. And so we can generate very large sets of easily measurable features, and we can ask whether or not we can predict which syllable the animal is expressing at any given moment in time based upon these features using standard deep learning techniques. And that works. We can't predict all syllables equally well, but we can predict some syllables perfectly that correspond to each of the major types of behavior, and we can predict about half of the syllables like 90% of the time. And that's more than good enough to do the thing that we want. And we can do that with very low latencies, so sort of circa 1 frame latency, so 30 millisecond latencies. So if the average length of a syllable is 300 milliseconds, knowing within 30 milliseconds that you're expressing a syllable this is fine. So given this gives us the ability to identify specific behavioral syllables, and on a specific behavioral syllable to trigger an optogenetic stimulus. So we're just starting this now, so I'm going to show you one panel of exciting preliminary data in which we picked a particular behavioral syllable that we can call with 95% accuracy, and we reinforced it via optogenetic provision of dopamine during the expression of just that syllable. And what we see is that if we look at just that syllable, as we do that over days, the expression of that particular syllable goes up, so we can reinforce specific behavioral motifs through the provision of dopamine in this closed-loop configuration.
What was it?
This was a rear. This was a rear. It was a rear. And you might imagine those are the easiest. It's the first one we did. Because the first principal component, actually this is an important point about mouse behavior in general, the first principal component's in Z. And so the use of 3D cameras to characterize mouse behavior is important because of that fact, and that's why we picked it. Okay? But if we do this, we can clearly reinforce individual behavioral syllables, And importantly, we can ask what happens to the other behavioral syllables. So if you look at the nearest neighbor syllables, this is the most important thing, nothing happens to them. That argues, I would say on the surface, that again, there's something right about our segmentation via MoSeq, because the brain, if this syllable were this syllable, you might expect that our reinforcement would generalize and bleed out across these different types of rears. And that's not what's happening. Instead, we're specifically reinforcing one pattern of behavior and observing no change in the other. So given that we're increasing the expression of one pattern of behavior, what happens to the others? What we find is that more distant behavioral syllables in general, broadly, become slightly downregulated to accommodate this increase. And Andres, we're looking right now at what this means for sequences. Obviously, our sequence probabilities themselves change. You can imagine doing a kind of temporal accounting here. Maybe there's also some change in prior syllables, right, or subsequent syllables that are sequence dependent and that we have to look at directly. So we're doing that analysis right now. That's of course what we care about. But what's interesting here is that there's this difference effectively between what happens here and what happens here. Almost like these syllables are being protected in some way from changing too much. And so we're thinking about what that might mean. But this is just meant as a proof of principle for kind of where we're going, which is to do closed loop manipulations of reinforcers, of punishments, and of neurons in the dorsolateral stratum itself to think about how neural systems support the generation of structured behavior. The last thing I'll mention is that we're extending beyond mice. So we have some preliminary data that suggests that MoSeq works in rats. With Hopi Hoekstra, we've begun doing some imaging in Paramiscus, other mouse species. And we've done actually not some insubstantial amount of work with multiple collaborators on different forms of human data. What you're looking at here is 7-axis accelerometer data on an autism spectrum disorder patient that's having a repetitive behavior. Okay, so that's what each one of these lines is one of these axes on the accelerometer. What you're going to look at as I play this movie are two different versions of motion sequencing in real time segmenting these data. And what you can see is that motion sequencing in this unsupervised way rapidly learns sensible looking behavioral motifs from this very simple accelerometer data. And that suggests that these kinds of time series methods for thinking about and organizing behavior might be generally useful, not just for thinking about mice, but for thinking about all sorts of other species, and perhaps even translating our understanding of behavior across species. And so with that, I will finally end. I just want to mention that this work was, the neural stuff that I showed you was primarily done by Jeff and Wynn, as I mentioned, in close collaboration with Bernardo Sabatini's lab. A lot of the initial development of MoSeq was done by Alex, Matt, and Ralph. That was in collaboration with Ryan Adams' lab. Currently, we're doing a lot of the model development in collaboration with Scott Linderman, who is in Liam's lab now, but is moving to Stanford in his own independent position. And we're planning on closely collaborating on model embellishments for the next several years. This is my lab, this is the funding, and I thank you for your questions.